our next topic is structure of Megasporangium. That is, we have to study the structure of ovule. So students, what we have studied so far? We have studied about the basic structure of a flower. We know the essential walls of the flower. The essential walls of the flower are androecium, that is the part of male reproductive system, that is stamen, and gynoecium, that is female reproductive system, that is pistils. We have also studied about the accessory walls. The accessory walls are the sepals and the petals, right? That is calyx and corolla. We have also studied about the structure of male reproductive part, structure of stamen. We have studied about the structure of anther. A typical anther is a dithecous anther. It is tetrasporangiate. We have studied the internal structure of the anther. We have studied about the anther wall, how dehiscence of the anther takes place. We have studied about the microsporogenesis, that is how microspores are formed. We have also studied about how pollen grains are formed. Pollen grains are male gametophyte. So we have studied that also, that how there is formation of pollen grains. We have also studied about palynology, that is study of pollen grains. What happens if there is allergy of pollen grains? Which pollen grains are allergic pollen grains? So we have studied about the male reproductive part of the flower. Today we are going to study about the structure of Megasporangium, that is the structure of the ovule, right? So we'll quickly revise. So we have studied about gynoecium. Gynoecium is a female reproductive part, right? It consists of multiple pistils. We know the structure of pistil also. It consists of a bulbous stigma, an elongated style, and a swollen structure that is called as ovary, right? This ovary is resting on the body. That body is called as thalamus or receptacle, right? So this is stigma, this is style, this is ovary, and these three structures together form pistil, right? So pistils are the part of female reproductive system, that is the wall of female reproductive system is called as gynoecium, right? A flower, a single flower can have a single pistil, then it is called as monocarpellary, or it can have multiple pistils, then it is called as multicarpellary. These pistils, which are multiple in number, are also of two types. They can be in the free form. Those are free pistils, which means there are many pistils but all the pistils are free. Or the pistils can be fused pistils. That is called as apocarpus, and fused pistils are together called as syncarpus, right? Then we also know that this style, right? Style can be of two types, right? So style can be of two types. The style can be a hollow style or it can be a solid style. The name itself says, say this is a style portion. If the style is peripherally bounded with a single layer of the epidermal cells and inside the style is hollow, such a style is called as hollow style. But if the style is completely filled with the cells, right? If the style is completely filled with the cells, such style is called as solid style. So there are two types of styles. There is a hollow style and there is solid style, right? So pistil is a part of female reproductive system. It consists of three major parts, that is stigma, style and ovary. What is present within ovary? Within ovary will be the presence of the ovule. So ovule will be present within the ovary. Say this is ovule, right? The arrangement of the ovule within ovary is called as placentation, right? So how is the ovule arranged within the ovary? That is called as placentation. You have studied different types of placentation in your 11th standard. So what is it? How ovule is attached with the ovary, right? There can be a single ovule or there can be multiple ovules present inside the ovary, right? So this is the basic thing. Gynoecium, female reproductive part. It consists of stigma, style and ovary. If a flower possesses a single pistil, it is called as monocarpellary. If it possesses multiple pistils, it is called as multicarpellary. If all the pistils are fused, it is called as syncarpous. If all the pistils are free, it is called as apocarpous. The style is also of two types, hollow style or the solid style. The name itself says if the style is lined by the epidermal cells but inside it is empty, such a style is called as hollow style. But if the style is completely filled with the cells, such a style is called as solid style, right? So now what we have to study? We have to study about the structure of the ovule. Ovule is also called as Megasporangium. Pause. So ovule is present within the ovary. The attachment of ovule within the ovary is called as placentation, right? So say this is the wall of the ovary. This is the wall of ovary. Ovule will be attached with the wall of the ovary through some tissue. So say this is the tissue through which ovule is attached with the wall of ovary. So this tissue is called as placenta. Placenta is a tissue which provides attachment of the ovule with the wall of the ovary. Coming to the structure of the ovule, ovule consists of two parts. The first part is a stalk-like structure and the second part is the body of the ovule, right? So it consists of a stalk and a body, right? So this part is called as the stalk of the ovule. It is called as funicle. It is called as funicle. The point where the stalk is attached with the body of the ovule is called as hilum. Hilum is the point of attachment between the stalk and the body of the ovule. What we will see in the body of the ovule, we will be able to see that the body of the ovule consists of layers, right? A typical ovule consists of 2 layers, right? These layers are called as integuments, right? We are studying the structure of a typical anatropous ovule. There are different types of ovules such as orthotropous, anatropous, hemianatropous, campylotropous, circinotropous. We will study it in the next lecture, right? But the ovule structure which we are studying is of a typical anatropous ovule which is commonly seen in angiospermic plants. This ovule is an upside down ovule. It consists of 2 parts, the stalk which is called as funicle, and the body part. The attachment between stalk and funicle is called as hilum. The body part possesses the layers, right? These layers are called as integuments. Anatropous ovule consists of 2 integuments. Hence, it is called as bitegmy. There are 2 integuments present. This one is the outer integument and this one is the inner integument. So these layers are arising from one place, right? That end of the ovule from where these integuments are arising, that end is called as chaliza. So the body of ovule is divided into 2 ends, chaliza, and the opposite end is called as micropyle end. So what is chalizal end? The end from where integuments are arising. And the other end where the integuments are not covering completely but there is an opening left out. That end is called as micropyle end. Right? What is present within ovule? Within this integuments, the integument is filled with the mass of the tissue. So this mass of the tissue which is present is called as nucellus, and each and every cell of nucellus is a diploid cell, right? So the ploid is diploid. So within integument is a mass of tissue, that is nucellus. One cell of the nucellus, which cell? The cell which is present towards the micropyle end. Say this cell is going to enlarge and it is going to function as MMC, that is megaspore mother cell. In the male reproductive system, you have also seen MMC. Over there, it is related to microspore mother cell. Here, it is megaspore mother cell. So, one cell of the nucellus which is present towards the micropyle end, the cell will be again diploid cell because all the cells of nucellus are diploid cell which is going to function as megaspore mother cell, and this megaspore mother cell is further going to give rise to an embryo sac. Embryo sac is a female gametophyte which possesses the female gamete, that is egg. This nucellus is also of 2 types, right? So if ovule possesses less nucellus, such ovule is called as tenuinucellate. If it possesses plenty nucellus, our next topic is formation of embryo sac. What is embryo sac? Embryo sac is a female gametophyte, right? So we have studied about the structure of ovule. We have studied the structure of a typical ovule, that is anatropous ovule. We have studied the parts of that ovule. We know where is megaspore mother cell present. Megaspore mother cell will be present towards the micropyle end, right? So it will quickly revise it. This is the stalk of the ovule and this is the body of the ovule. Body of the ovule possesses funicle. Body of the ovule possesses layers in the form of integuments, and within it there will be the mass of the cell. That mass of the cell is called as nucellus, and one cell of the nucellus, so this end is chaliza end, this end is micropyle end, and one cell of the nucellus towards the micropyle end is going to function as a megaspore mother cell. This we have studied, right? Today we have to study about how there is formation of an embryo sac. So how this megaspore mother cell is going to give rise to a female gametophyte, right? So we know, say this is a diploid megaspore mother cell. This undergoes one meiotic division. We know meiosis is a reductional division. So from a single cell, what is formed from a single cell? A cluster of 4 cells are formed, and all the 4 cells will be haploid cell. So this single megaspore mother cell gave rise to 4 haploid megaspores, right? This is micropyle end and this is the chalical end. Now all the 3 cells which are present towards the chalical end, so these 3 cells are going to get degenerate, right? So once they starts degenerating, say the cells are degenerating, so what is left? What is left is only a single haploid megaspore. So this is a single haploid megaspore which is present towards the micropyle end, right? Now what will happen with it? This single cell undergoes 3 successive karyokinesis. So there will be 3 successive karyokinesis. So let me enlarge it. Say this was the cell. It had a single nuclei. Now we are talking of karyokinesis, which means there will be division of only nucleus. There will not be any cytoplasmic division. So there will be one karyokinesis, and this 3 successive karyokinesis will be of which type? It will be of mitotic type, right? So there will be mitotic karyokinesis. So a single nucleus is going to give rise to 2 nucleus, that is first karyokinesis, right? So single one gave rise to 2. This is the first karyokinesis. This 2 by the second karyokinesis gives rise to 4 nuclei, and by the third karyokinesis, how many total nuclei are formed? The 4 are again going to give rise to 4 more. So total 4 nuclei are formed in this single megaspore cell. Clear? Now what will happen? Now this nuclei are going to undergo movement, right? So 3 nuclei will move towards chalical end and 3 nuclei will move towards the micropyle end. So let me again draw it. So 3 nuclei will move towards the chalical end, 2 nuclei are present in the center, and 3 nuclei will move towards the micropyle end. This karyokinesis is followed by cytokinesis. So there will be formation of cell wall, plasma membrane, right? So how will it happen? Right? So over here there were 3 nuclei, in the center there were 2 nuclei, and again in the micropyle and also there are 3 nuclei. 3 nuclei in the chalical end, 3 nuclei in the micropyle end, and 3 nuclei in the center. Now this nuclei will undergo, there will be cytokinesis, but cytokinesis will occur only towards the chalical end and micropyle. So there is formation of cell wall and plasma membrane. Similarly at the micropyle end also there will be formation of cell wall. These 3 cells which are formed towards the chalical end are called as antipodal cells. In the center, 2 nuclei are left as it is. There is no cytokinesis around them. These 2 cells which are towards the micropyle end are called as synergid cells. In the center, there is one large cell that is called as egg cell, right? So this cell is actually the egg cell, right? This is egg cell. And this synergid and the egg cell together forms egg apparatus. This is clear. So this is the structure of embryo sac. So we will draw the structure of embryo sac, an enlarged structure of embryo sac. This embryo sac developed from a single megaspore mother cell, right? How many nuclei are present in it? We know that 3 nuclei are present towards the chaliceal end, 3 nuclei are present towards the micropyle end, and 2 nuclei are present in the center, right? So total there is the presence of 3 plus 3 plus 2, that is 8 nuclei are present. We know that there was cytokinesis at the chaliceal end as well as at the micropyle end. The cells towards the chaliceal end are called as antipodal cells. The 2 cells towards the micropyle end are called as synergid. The center cell is the egg cell, and together they form egg apparatus, right? And in the center there are 2 polar nuclei. So this is the structure of a typical embryo sac. This embryo sac is called as monosporic type of embryo sac. Why? Because it developed from a single megaspore mother cell. It is how many nucleated? 8 nucleated. How many cells are present? 1, 2, 3, 4, 5, 6, and this large cell, 7 cells. So it is 7 cell, 1, 2, 3, 4, 5, 6, 7, 8. 8 nucleated. It is a polygonum type of embryo sac because it was first discovered by the scientist Hofmeister in Polygonum plant. That is why it is called as polygonal type of embryo sac. So embryo sac is a female gametophyte because it possesses the female gamete, that is egg cell towards the micropyle end, right? This entire process is called as megasporogenesis. So from a single megaspore mother cell undergone meiotic division to form 4 haploid megaspores. 3 megaspores towards the chaliceal end degenerated. Only 1 megaspore towards the micropyle end left. This haploid megaspore underwent 3 successive karyokinesis. So with each karyokinesis, there is replication of the nuclei. It is mitotic karyokinesis. Ultimately, 8 nuclei are formed. Then there is movement of the nuclei. 3 nuclei towards the chaliceal end, 3 nuclei towards the micropyle end, and 2 nuclei remains in the center. The nucleus towards the chaliceal end and micropyle end, there will be formation of cell wall around them, that is cytokinesis. So the cells, 3 cells towards the chaliceal end are called as antipodal cells. The 2 cells on the either side of the micropyle end is called as synergid cells, and the center large cell is the egg cell. The egg cell and synergid cell is together called as egg apparatus. So this is an embryo sac. It is a monosporic type. Why? Because it developed from a single megaspore mother cell. It is 7 celled and 8 nucleated type, and it is a polygonum type because it was discovered by Hofmeister in the Polygonum plant. In the next lecture, we will study about other different types of ovules. We have studied about a typical ovule, that is anatropous ovule. Our next topic is types of ovules. Right? So students, we have studied about the structure of a typical ovule, that is anatropous ovule, which is commonly seen in the angiospermic plants. It is an inverted ovule. We have also seen, right, that within this ovule how an embryo sac is formed. What is embryo sac? Embryo sac is a female gametophyte, which means it possesses the female gamete, that is an egg cell. How is this embryo sac formed? It is formed from a single megaspore mother cell which undergoes first meiotic division, and then there is 3 continuous karyotic mitosis followed by cytokinetic divisions, right? We have seen a typical embryo sac structure also, right? So next topic is types of ovule. So first of all, let us recollect the structure of the ovule which we have seen already, right? The typical ovule which we have studied is anatropous ovule. Anatropous ovule. Anatropous ovule is also an inverted ovule. This anatropous ovule is commonly seen in angiospermic plants. We have studied the structure of this ovule also. We know that ovule is connected with the wall of the ovary by a tissue. That tissue is called as placenta, right? We have studied that the ovule consists of 2 parts. One is the stalk part, which is called as funicle, and the another is the body part, right? So this stalk part is called as funicle. The connection between the stalk and the body is called as hilum. The body part consists of 2 ends, that is micropyle end and chaliceal end. In a typical anatropous ovule, the micropyle end lies towards the lower side, and over here lies the chaliceal end. How had we decided this ends? So we know that a typical ovule, angiospermic ovule, is a bitegmic ovule, which means it possesses 2 integuments. The end from where the integuments are arising is called as chaliceal end, and the end where there is an opening, a tiny opening, is called as the micropyle end. On the inner side of integument, right, there is the mass of tissue called as nucellus. From this cells of nucellus, a single megaspore mother cell is going to arise towards which end? Towards the micropyle end. And this single megaspore mother cell is finally going to give rise to an embryo sac. So this structure is embryo sac. We know it is 7-celled and 8 nucleated. There are 3 antipodal cells towards the chaliceal end, 1 egg cell and 2 synergid cells towards the micropyle end, the large central cell. So total there are 7 cells and there are 8 nuclei, because there are 2 centrally placed nuclei which does not undergo cytokinesis. Those are called as polar nuclei. So this is the first type of ovule, the structure of which we have already studied. It is an anatropous ovule. In anatropous ovule, chaliceal end is towards the upper side and micropyle end is towards the lower side, right? The second type of ovule is orthotropous ovule. This ovule is an upright ovule. In orthotropous ovule, all the 3, that is funicle, chaliceal end and micropyle end, all lies in a same plane, and it is an upright ovule. So let me draw it. This is the funicle of the ovule, right? So funicle is attached with the wall of the ovary by a tissue. That tissue is placenta. So this is placenta. This is funicle, right? And this is the body of the ovule, right? And this are the integuments. So micropyle end is towards the upper side and chaliceal end is towards the lower side. It is reverse of the anatropous ovule, right? That is funicle, and within it again there will be an embryo sac. So this is orthotropous ovule. It is an upright ovule, right? Anatropous ovule is commonly seen in angiospermic plants. Orthotropous ovule is commonly seen in gymnospermic plants, and gymnospermic plants are usually unitegmic. Angiospermic plants are usually bitegmic, which means there are two layers of integuments, and this are unitegmic, which means there is single layer of integument, right? So this is the second type of ovule, that is orthotropous ovule. Anatropous ovule is an inverted ovule with micropyle on the lower side and chalaza on the upper side. And orthotropous ovule is an upright ovule where funicle, chalaza and micropyle, all the three lies in the same plane. Micropyle is towards the upper side and chalaza is towards the lower side. This much is clear, right? The third type of ovule is called as hemianatropous ovule, right? So the third type of ovule is hemianatropous ovule. The name itself says, right, anatropous ovule is a completely inverted ovule. Hemianatropous ovule, so it is anatropous, but it is partially anatropous, which means hemianatropous ovule is a transverse ovule, right? How is the arrangement of it? We'll quickly see it. So this is the funicle, right? The stalk-like structure, that is funicle, or let me draw it like this. Yeah, so this is funicle, right? And the body of the ovule lies at the right angle to the funicle, right? So this is the body of the ovule, right? Again, these are the layers of integuments. Inside there is embryo sac, right? So this is body of the ovule. This end is micropyle end and this end is chalazal end. So which angle is formed? The funicle and the body lies at the right angle to each other. Such type of ovule is called as hemianatropous ovule. It is a transverse ovule. The body of the ovule is transverse. In anatropous ovule, the body of the ovule is inverted. Whereas in orthotropous ovule, the body of the ovule is upright ovule, right? Then the next type of ovule fourth type is called as campylotropous ovule. This ovule is in between the anatropous ovule and the hemianatropous ovule. So anatropous ovule is completely inverted. Hemianatropous ovule is completely transverse. So in between it, right, the ovule which lies at an angle in between the anatropous ovule and hemianatropous ovule, that type of ovule is called as campylotropous ovule, right? So this is funicle. So this is funicle. If it would be hemianatropous, then the body of the ovule should be right angle. If it is anatropous, then the body of the ovule should be completely inverted. But in this case, the body of the ovule is neither at the right angle nor it is completely inverted, but it is in between it, right? So neither it is 90 degrees nor it is completely inverted, right? So this is the angle which is formed, right? And inside there is embryo sac development. Such type of ovule is called as campylotropous ovule. This end is chaliceal end, this end is micropyle end. Clear? So campylotropous ovule is in between the hemianatropous ovule and anatropous ovule. The fifth type of ovule is amphitropous ovule. Amphitropous ovule, the structure of amphitropous ovule is similar to that of campylotropous ovule. The only difference is that in it, the embryo sac is horseshoe shaped of embryo sac, right? So first let's draw the structure of campylotropous ovule. This is the funicle and this is the body of the ovule. In it, the embryo sac is normal oval structure, whereas in case of amphitropous ovule, the embryo sac also becomes horseshoe shape. Other things are same, right? So this end is chaliceal end, this end is micropyle end, and this is the funicle, right? So such type of ovule is called as amphitropous ovule, where the only difference between campylotropous and amphitropous ovule is that the embryo sac is horseshoe shaped type of embryo sac. And the last ovule is Surendranagar ovule, right? This ovule, the funicle of this ovule is elongated and it makes a complete turn. So actually the body of the ovule is an upright ovule. Let me draw it first, right? So this is the funicle of the ovule, which is an elongated structure. And from here arises the body of the ovule. Present within it is the embryo sac. This is micropyle and this is chaliceal end, right? So the funicle is elongated and makes a turn. Such type of ovule is called as Surendranagar ovule. This is clear. So these are the 6 types of ovules. But the typical ovule, which is commonly seen in angiospermic plants, is an anatropous ovule. It is an inverted ovule, right? We know the parts: the funicle, which is a stalk, and the body. The 2 ends, micropyle and chaliceal end. The connection, the point of attachment between the funicle and the body is called as hilum. All the ovules are attached to the wall of the ovary through a tissue, that is placenta, and there are different arrangements of the attachment of ovule with the placenta, that is called as placentation, which you have already studied in your 11th standard. So we have to study the structure of the embryo sac, the diagram which is given in your books, and it is called as female gametophyte. Why? Because it is a gametophyte, it is a structure which itself possesses the female gamete, that is the egg cell, right? So it is monosporic type because it developed from a single megaspore, that is monosporic type. It is polygonum type because it was discovered by Hofmeister in the polygonum plant. It is 7 cells. We have counted the cells also: 3 cells towards the micropyle end, 1 egg cell, and the 2 synergid cells. 3 cells towards the chaliceal end, that is the antipodal cells, and 1 large central cell. So total it is 7 cells, and it is 8 nucleated, right? So all the 7 cells possesses nuclei actually. So actually this 3 cells and this 3 cells possesses nuclei, and there are 2 centrally placed polar nuclei. These are called as polar nuclei. Clear? Okay. So micropyle possesses a large egg cell. It is oosphere. It is a haploid cell, which means it is a cell which possesses a single ploidy of the nucleus. It is surrounded by the two cells. Those cells are called as synergid cells. So these two cells are called as synergid cells, right? What is the peculiar feature of synergid cells? This synergid cells possesses filiform apparatus. What is filiform apparatus? Those are the finger-like projections which arises from the cell wall of the synergid cell. So those are the finger-like projections which arises from the cell wall of the synergid cell, and they extend into the central cell, right? What is the function of it? To distribute the nutrients in the embryo sac. That is the first function. And second is it guides the entry of the pollen tube. We know that when there is pollination, which means when pollen grain comes and rests on the stigma, right, there is germination of the pollen grain, there is formation of the pollen tube, and this pollen tube is going to enter. The pollen tube contains male gametes. This pollen tube is going to enter inside this embryo sac. Usually it enters from the micropyle end, and who is guiding the entry of the pollen tube? The entry of the pollen tube is guided by this filiform apparatus, which is the part of synergid cell. Two centrally placed polar nuclei. This is a large cell. It is called as central cell. It is the largest cell of the embryo sac. And secondary nuclei, which means this polar nuclei which are present in it, forms the contact with all other cells through the plasmodesmatal connections, right? We know plasmodesmata, those are the modifications of the plasma membrane, right, which are connecting the cells. Chalazial end possesses the three antipodal cells, right? There will be one more thing which you have to remember is that cytoplasm is shifted more towards the chalaza, and micropyle end possesses two large vacuoles, right? So there will be presence of two large vacuoles towards the micropyle, right? So this is the structure of an embryo sac. It is a typical embryo sac. Clear? Coloring.